Perfect. Okay.
Hopefully that's full screen. Okay, wonderful. Thanks so much for having me. I'm going to start my timer. Um, Great to be able to share with you some of the work going on in my group. I usually start with these images where you can go left to right, and these are true images of phages that we've used in emergency patient treatment. And what you'll learn is that phage therapy is sort of resurged as an idea because we're running out of other options to deal with antibiotic-resistant infections, but we take an evolutionary lens to it. So I'll try and quickly go through how we approach phage therapy and the success we've had in the clinic with it. First thing I have to do is give a quick disclosure that I'm co-founder of a company that seeks to commercially develop phages for therapy. And here's the overarching problem, which I probably don't need to spend too much time on for this audience. Bacteria and bacterial infections have been a problem with humans as long as humans have been around. We capitalized on using antibiotics, but they don't work so well anymore. And this is a global problem. So if you look at projection maps, we could talk about how accurate a map like the upper right is, and it's not so much the accuracy of it that matters. It's a global problem where there's going to be a huge effect on human mortality. And the problem associated with this is that big pharma doesn't pay as much attention to it. There's not as much money in it. So when antibiotics go to market and they're sold, that's fine. But many of the ones that are discovered more recently are last resort drugs, and they typically sit on the shelf and they're not really sold. So this has created what's called a discovery void. We can take a step back in time and look at old technology that was very successful in the early 1900s, soon after phages were discovered. These are viruses that are specific to killing bacteria if they have what are called lytic phage reproduction cycles, as shown in the cartoon in the middle. And as I said, this has been attractive ever since phages were found. And one of the co-discoverers happened to have a Yale appointment way back in the day for a short period of time in the early 1900s. But the point is primarily this work has been used and developed with lytic phages, which are lethal to bacterial targets. And the bonus is that they're self-amplifying drugs. So they make copies of themselves as they replicate in cells and exit them to infect and kill cells of similar type. But there's an early phage therapy problem that still remains to some extent a concern today, is that lytic phages tend to be very specific, and they'll often kill only a subset of genotypes, even in a single target bacterial species. This specificity is attractive because it gives you good targeting without off-target effects, but it's certainly a concern that you can or cannot, and often you cannot, use this technology widely across genotypes, as shown in this cartoon on the lower right. If you consider strains 1 through 10, and you might have several phages that you'll bundle together as a cocktail to try and cover as much of the strain diversity. And yet you'll see, for example, strain 4 and strain 5 are completely missed anyway. So cocktails have been an attractive idea, but it doesn't mean that they'll work to cover all of the target strains that you want to cover. Now we come to more of the modern day, and to me, I'm just going to quickly say that I don't think host specificity is as big of a problem as we consider it to be, because on the left, this is what's called a killing grid, where you take phages where we know them and we know their genomes, and we take some clinical strains that presumably they've never seen before, because the strains come from the clinic, whereas the phages come straight from nature. And everywhere you see in this grid a dark blue square filled in is where the phage on the left killed the challenge bacterium above it in that column. So this means that you can find naturally broad host range phages, and the idea is that you would ideally use these in therapy. But even more recent technology could be, assembling together from bits and pieces of DNA and sort of taking a Frankenstein approach of building the phage that you want if you know the amino acid substitutions that give it broader host range. So we've been working with partners such as New England Biolabs in order to develop that technology and test it. Here's the bigger problem to me as an evolutionary biologist. I would predict that every time that you use phages in therapy, you're going to select for bacteria to evolve resistance to them. And ever more so, we know this from cartoons like in the lower left that remind me to say that a vast number of phage resistance mechanisms have been known for some time, and it's almost a cottage industry now for some excellent laboratories around the world to find them almost on a weekly basis, new ones. So there's a lot that's stacked against phages in terms of their ability to infect bacterial hosts, and evolution can make them even better defended against these phages. So how are you going to address this problem? Two main ways. One would be somehow minimizing the evolutionary potential of the target bacteria so that they don't evolve resistance. I think that's a hard thing to achieve. And instead, we are looking at the second bullet point. If we just assume that therapy is going to select for resistant bacteria, can we steer the evolution down a path that we prefer? And that's described here in this cartoon, where the first idea we put forward in this kind of bringing evolution to phage therapy and updating it for modern times. Think of first this yellow triangle, where many bacteria that are problematic in the clinic, they have complexes of proteins called efflux pumps. And when antibiotics enter the cell, they're actively pumped out, and this can give bacteria resistance against different drugs drawn from different drug classes, very broad resistance to antibiotics. So what we put forward is that if you have a phage that is able to bind to these efflux pumps, then it will kill the bacteria in the classic sense of phage therapy, but it will also exert selection pressure along this yellow arrow so that the bacteria are ideally backed into a corner where they solve the phage problem by evolving resistance, but they compromise functionality of something that's important in pathogenicity. And in this case, it's an efflux pump. So this means that the bacteria have gained resistance to the phage, but they've become resensitized to antibiotics. And a faster description would be in the purple portion of this purple triangle, where if you have a shield or a capsule around these bacteria that cause them to be not detected by your immune system cells, you find a phage that is specific to binding to those target bacteria by binding to the capsule, kills the bacteria, but also selects for those that have lost the capsule and they're more susceptible to destruction by your immune system cells. So the portion in yellow, I should emphasize, is an old thing that is seen in the literature, although mechanistically it was not clear why. It's an example of how phages and antibiotics can synergize to better kill bacterial populations. So we are leveraging such technologies as in yellow because we especially prefer to develop phage therapy alongside antibiotic use. Still, the standard of care in many clinical settings is to deploy antibiotics, even if they work poorly. So we would like to have this synergy to our advantage when we deploy the phages. We've taken some theoretical exploration to this with partners such as Joshua Weitz, who's now at University of Maryland, College Park. And we were wondering why with one dose, essentially we could find in 2018 a case that we published on a man who had an aortic arch replacement that was infested with multi-drug resistant Pseudomonas aeruginosa, and we were able to destroy that infection when antibiotics could not. And the main point of this theoretical exploration is to say that you can have three things working to your advantage: an antibiotic and a phage synergizing like I talked about. But on the lower right here, if the individual has an active immune system, sometimes it's a matter of dropping the bacterial load down enough that the immune system can participate in clearing the infection, and you get this big white space of expected efficacy when you have all three synergizing together. So how we deliver these phages, I'll talk about as I finish up to give you some clinical evidence. It depends on where the infections are happening in the body. So we've looked at aortic arch replacement, as I mentioned, also plenty of prosthetic joints to deliver therapy to patients for which antibiotics are not working, and we use this technology in an emergency. I'll talk in a moment about inhaled delivery of phages directly to the lung to try and treat pulmonary infections. We have not been doing much with systemic infections of the human body, like sepsis, but there you might deliver phages directly to the bloodstream because you're trying to go after a bloodstream infection. So looking at a case series that we are in the process of finalizing for publication, in this case, we have 9 individuals with cystic fibrosis who were born with a genetic condition that leaves them more vulnerable to pulmonary infections, and those are increasingly antibiotic resistant. So with our team and our Center for Phage Biology and Therapy, in particular for these 9 cases, we used inhaled therapy to target Pseudomonas aeruginosa that was multi-drug resistant. And I'll just highlight one of the phages that we used when I finish up by showing the clinical data. We look for phages that have excellent properties to kill the bacteria, but also to exert these trade-offs or steer the bacteria down an evolutionary path that we prefer. So very briefly, I'll introduce a phage called H6, which binds to the type 4 pili of Pseudomonas aeruginosa. And why would you care? It is because these bacteria use the pili to sit down on cells, on your cells to form biofilms. And this is problematic in infected organs like the lung because this can occur alongside the bacteria secreting virulence factors like piocyanin, and this will destroy lung tissue and ultimately lead to either a lung transplant or pulmonary failure. So the idea is if we deployed a phage like this in patients who need the therapy and we use this particular phage, do we not only destroy the infection or at least reduce it, do we also select for bacteria that have become compromised for these properties and become less pathogenic as a trade-off? So on the left, we'll see that in these 9 patients, if you look at the amount of target bacteria in their sputum coming from their lungs, before therapy, there's a very high bacterial load. And after therapy, in all cases, the load drops. That shows us the phage therapy is working. And on the right, the clinical benefit is that the lung function improves as measured with something abbreviated as FEV1. And this is how much air can you breathe out of your lungs when told to do so by the physician over the standard period of time. And across all 9 patients, there was this improvement. You will note that some of these patients were very, very bad off in terms of their lung function to begin with. And we're mostly trying to avoid them from having to undergo a lung transplant. So to finish up, do we see evidence that in these treated patients, not only did I quickly show you that the therapy works, does it steer the bacteria down an evolutionary trade-off outcome? So when this phage is used, for example, in patient 2, we find that pre-therapy, you can isolate clones from sputum and look at the ability of those clones to sit down on cells of human origin in the laboratory and start to form biofilms. And they're variable for this across clones, but they're quite good at doing it. After therapy or post-therapy, this troughs out because in this case, we believe we're successfully killing the bacteria, or we're also selecting against the presence of these type 4 pili. Related to that, the pili are functioning in the secretion of piocyanin as a virulence factor, and we also find that in these 3 patients, 2, 7, and 8, post-therapy, piocyanin production drops off in these bacteria, and you can also track in a single patient through time the loss of piocyanin production. So all this makes sense in terms of exerting the trade-offs alongside using the therapy. And one more thing about this is that the secreted virulence factors, piocyanin, your cells are not happy when they see piocyanin, and this causes a lot of inflammation. So one would think if we take clones post-therapy and we look at them in the laboratory and study them for inflammation markers like IL-8 and IL-6, indeed we observe that post-therapy, the cells of human origin that are exposed to these clones post-therapy, they are not undergoing the inflammation response as when the wild type bacteria are sitting on those cells. So this is, again, going in the right direction. So I'll end with saying that this very busy slide is to illustrate that we can look at clones taken from patients, look at SNPs or single nucleotide polymorphisms in the expected target loci to look at the genotype-phenotype mapping of when we get these phenotypic trade-offs and the clinical improvement because the patients are doing better and the bacteria may still be there, but they're less pathogenic. We can also pinpoint exactly the mutations responsible. So that was a whirlwind tour, and I hope that I didn't go through that too quickly. I'd like to finish by acknowledging the amazing people who've worked with me in my career, in particular, providing some of the data that I showed today. So if